안녕하세요. 제 이름은 장우영이라고 하고요. 제가 한국에 돌아와서 김민식 교수님, 디지스트에 계신 김민식 교수님과 질량 분석기를 통해 다양한 연구를 했는데, 그 중에서 작년 후반쯤에 그 자폐증에 대한 논문이 나와서 그거를 발표하려고 이 자리에 나왔습니다. 오늘 발표는 영어로 진행을 할 거고, 어, 어, 발표 타이틀은 Cntnap2-dependent molecular networks and autism spectrum disorder revealed through an integrative multiomic analysis입니다. So what is autism? Uh, Autism is a group of complex neurodevelopmental disorder characterized by repetitive behaviors and characteristic patterns of behaviors with difficulty with social communication and interaction. A person with ASD often shows impairment in socialization, impaired communication, repetitive patterns of behaviors. Have you seen the movie Marathon or the TV show Woo? In these media, they highlighted the difficult challenges the autistic patients have to face day to day. But also, the patient disorder affects the people surrounding them. This is highlighted in a quote, "My wish is to live just one more day than Chon." In the US, approximately 1 out of 54 children are diagnosed with autism spectrum disorder. Boys are 4 times more likely to be diagnosed with autism than girls. In Korea, the prevalence rate of ASD is around 3%, and one issue about ASD prevalence is the fact that prevalence rate keeps increasing every year. This might be due to the increased marital age or neurotoxic environment factor or updated criteria for ASD diagnosis. Anyways, increasing ASD prevalence is going to have a huge impact on society near future. But despite this, the significance of the rising number of the cases, our understanding of ASD is very poor. Still, we don't know what causes ASD. Therefore, there is no current available diagnostic method or cure. Currently, therapy for ASD is targeting symptoms, not ASD itself. In the beginning, the genomic field was the first to identify patients' unique genomes, and their ASD susceptible risk gene resulted in ASD genetic mouse models. And by using these ASD genetic mouse models, the molecular mechanism of ASD in various omics field can be explored. And in the slide, I brought an example of a database, or one of the largest database for autism spectrum disorder called Safari genes, along with a few of the syndromic genes that represent autism. Before genomic success, it was hard to study the molecular mechanism of ASD in many omics fields due to the fact that the origin of the pathological sample is the brain itself. The susceptible risk gene for genomic study helped to produce ASD genomic mouse model. Through genomic research, we can now explore the molecular mechanism of ASD in mRNA, protein metabolomics, using ASD mouse models. With the help of Dr. Jiwon Park, the core biological process of ASD was collected from the available brain proteome database of the various ASD mouse models. From the literature, we found 7 ASD mouse models, D7, and including our 2 data regarding the autism mouse models, we have proceeded with the meta-analysis. Through meta-analysis, 6 major processes commonly shown in multiple ASD mouse models were explored. It was mitochondrial dysfunction, cell-to-cell interactions, and vesicle transport, neuron development, and ion homeostasis and protein translations. So to explain what Cntnap is, in 2006, Cntnap2, a recessive mutation, was found in a family where a family suffered from cortical dysplasia focal epilepsy syndrome. However, the family has also represented a lot of ASD symptoms, such as intellectual disability, language regression, and epileptic seizures. Because of this, Cntnap2 was associated with potential ASD marker back in 2006. We can also see that Cntnap2 acts as contactin between the synaptic membrane to the postsynaptic membrane, and its functions are known as cell-to-cell adhesion molecule in the interneuron. However, recent study also revealed that Cntnap2 and Cas in the presynapse membrane binds and functions as glutamate vesicle. The full understanding of Cntnap and its direct and indirect function is still unknown. In collaboration with Professor Young-Suk Lee in Seoul University, we confirmed that the Cntnap2 knockout ASD mouse model was impaired in social preference. As seen in the right, mice with the Cntnap2 knockout showed low social preference, and they have lost their natural preference for unknown mice. After the Cntnap2 knockout mice behavior test, we dissected the medial prefrontal cortex of the control and the Cntnap2 knockout mice and performed filter-aided sample preparation, fast protein digestion method using tryptic enzyme. The sample were labeled with tandem mass tag, and the samples were fractionated to reduce the complexity of the peptide variant. The LC-MS-MS analysis by data-dependent analysis method was performed using Lumos fusion orbitrap mass spectrometry from Thermo Fisher Scientific, and we obtained 8,821 proteins and identification of 107,644 peptides through proteomic profiling, and we found 884 differentially expressed proteins, where we call DPs, between groups. The DPs were noted when two or more D-peptides were identified as DPs, and D-peptides were identified when student t-test value showed equal or less than 0.05 and median ratio test a value of less than 0.1 value. And using this 884 DPs, four different molecular processes categorized were identified: mitochondrial dysfunction, vesicle transport, neuronal development, and metabolomic processes. And as we see here, only the metabolomic processes seem to show up strongly in the upregulated section, while the other showed in the downregulated DPs. Among the DP, one of the most prominent upregulated protein was shown in metabolic processes. Further, through Cntnap2 mouse high radical geo-analysis, we found 40% of the proteins were involved in metabolic processes, which suggests unimportance. So as metabolomic alteration was one of the major processes in proteome, thus we conducted a targeted metabolomic analysis. Similar to proteomic analysis, after the behavior test, Cntnap2 knockout mice, medial prefrontal cortex was collected. After the homonization and centrifugation, the supernatant, 10 microliters of it, was labeled with the absolute IDQP 180 kit from BioCrate with the internal standard, and then further it was derived with the phenyl isothiocyanates labeling reagents for amino acids. Half of the samples were used for LC-MS-MS, while the other half was used for flow injection analysis with mass spectrometry using targeted mass spectrometry, MRM, and we have obtained 114 metabolites. And among those, through our reanalysis, we have found 7 metabolites differentially expressed between control and Cntnap2 knockout. To highlight a few, phosphatidylcholine 38 and O and O 42 and 2 shown to regulate the axon skeleton. Specifically, our suggested mechanism shows MAP ERK pathways was deactivated, leading to an increase in lipid metabolism. Also, sphingomyelin, D18, 1, and 20, and 2 might be potential biomarkers for ASD diagnostic, for they have only expressed in Cntnap2 knockout mice. Through proteometabolomic analysis, we found Cntnap2 associated signatures. But one issue with mouse model is some of the results from mice model are not comparable to those from human due to the organism differences. To address this, we have used publicly available post-mortem ASD patients' brain omic data was integrated with our Cntnap2 results. And using our proteome and metabolomic data from Cntnap2 signatures was compared with the transcriptomic, lipidomics, and metabolomics of the ASD and the control. And we tried to find co-expressed molecular pathways. And we have used PFC omics data of the bulk RNA sequencing data, including 38 healthy and 25 AST, which is here, and and 40 healthy and 32 AST PFC here for metabolites, and 403 healthy and 50 AST PFC, and shown in here with the lipids, were analyzed with the same parameters. And we obtained 48 genes that overlap with the genes and the protein expression patterns, and 2 metabolite that shows overlapping patterns shared commonly in ASD patients and Cntnap2. That means that even though we identify 7,000 or 73 overlapping genes and metabolites, we had to have the same expressional patterns between them. And we found, and to talk about a few of these, we found 35 downregulated genes were significantly associated with synaptic vesicles functions, including synaptic vesicle, synaptic membrane, and secretory vesicle, and neuronal axon development, and axon terminus, and neuronal projection. And these functions are pivotal for maintaining synaptic functions and neuronal migration, and their dysregulation can lead to synaptic dysfunction and neuronal migration deficit, which can be observed and have been linked to Cntnap2 mutation and other neuronal development disorders such as ASD and intellectual disability. And the 5 downregulated genes highlighted in the right here are ASD syndromic according to the Safari database. And 12 upregulated genes were found mainly involved in mitochondria, lipid metabolism, by including mitochondrial lipid metabolomic processes, fatty acid beta oxidation, which were also known to be associated with ASD. Next, to map specific network to cell type directly affecting by Cntnap2, we integrated public forebrain organoid single-cell RNA sequence data from Cntnap2 mutation carrier of ASD patient. First, we selected PFC-specific cell types among all the cell population in the organoids by comparing the in situ hybridization data of tissue from Allen developing mouse brain atlas using FoxHunt. The best correlation was observed from embryonic mouse day E15, highlighted in the red box, and among the 5,932 cells of the 392 pallium specific cell, and among the total of 28,108 cells in organoids were mapped with 2 dorsal pallium, as we see on the figure B and C. And we confirmed that dorsal pallium develops into the PFC, therefore 5,932 cells were further analyzed. And using these cells and Lydian cluster method, we identified 13 clusters, and of which 5 representative cell types were curated using neocortex markers of excitatory neurons, interneurons, neuronal progenitor cell, radioglial cells, and ambiguous cell. And to find out ASD-associated challenges at the cell type level, we performed DG analysis by comparing each cell type cluster in the ASD organoid with other Cntnap2 proteome R experiment, and Cntnap2 integrated with ASD patient overlap data, which we just showed, and healthy normal organoid with cell type specific DGs. In our cluster analysis, excitatory neurons showed more correlation with Cntnap2 knockout and Cntnap2 integration right here, excitatory neuron, and um, sorry. And when looking at the organoid by itself, we saw that interneuron and radioglial cells show correlation with Wnt signaling and cell-to-cell signaling by Wnt and neuronal progenitor cell shown in TOR signaling pathways. And looking at the UMAP that express Cntnap2 expression in healthy NASD, Cntnap2 was generally expressed only in excitatory neuron progenitor cell cluster, along with excitatory. However, Cntnap2 was differentially expressed between ASD and control sample only in excitatory cluster, not in the neuronal progenitor cell, suggesting that Cntnap2 is excitatory neuron have strong contribution to ASD development. Right here, excitatory shows difference, while neural progenitor cell doesn't. When looking at the excitatory specific DGs, shown similar expression patterns to Cntnap2 knockout proteome data, we see that right here, and we made a string map using string database, and Cntnap2 showed a high hubness of p-value of 0.0767, indicating a strong association with Cntnap2 in both Cntnap2 knockout DPs and excitatory specific DGs from the single cell organ. ASD molecular network associated with Cntnap2 was created by Cntnap2 knockout mouse MPFC. And proteometabolomic analysis, DEPs, DMs, and human integrated DGs, and Cntnap2 defect organoid single cell profiling. And by integrating them, a total of 112 DEPs or DGs and 6 DMs were enriched in lipid metabolomics, mitochondria, synaptic vesicle, and exon structure from our mouse data and from single-cell RNA organoid data, and 71 excitatory specific DGs involved in synaptic function, neuron projection, and mitochondria. To understand ASD molecular mechanism linked to Cntnap2, we generated a hypothetical molecular network model with selected molecular alteration and myelin sheath and mitochondria, synapse, and neuronal projection. These is our mechanism figures. The bigger the knot represent genes that show same expression patterns with Cntnap2 and human autism patient, knockout and autism patient, and the node color of green to red represent Cntnap2 knockout expression, while the outer layer shows ASD organoid DG's expression. As seen in our previous metabolite data, we observed that lipid metabolism involved in gene has been regulated in increase right here, and we suspect that an increase in acetyl-CoA. And also, we see that mitochondrial complex 2 Q coenzyme affecting proteins has also been increased here. However, for this function to work, they need calcium, but MCU gene, which is the main calcium intake of the mitochondria, has been downregulated. A strong expression of ATP despite the lower calcium intake has been led to involvement of reactive oxygen species. Further, there have been suggestions that mitochondrial dysfunction in calcium intake will create a charge defect through a neuron, leading to irregular synaptic pulse, which is found in Cntnap2 knockout mice of the irregular pulse. And another vesicle that is strongly related to autism, and which is also shown in our all of our data, is synapse. And specifically, we're going to talk about synaptic vesicle transport system. And we see that the main co-expression genes are downregulated, such as RAP35 and septin-6 and such. However, nectin and Cntnap2 cell-to-cell interaction proteins were also downregulated, and which we expect the reduction of the efficiency of the endosomal and exosomal activity, which will lead into uncontrolled or irregulatory and synaptic input, resulting in irregular action potential as well. Final pathway that we shown is that the neuron projection, the cytoskeleton organization and its regulatory pathway molecules were decreased. Cntnap2 is known to regulate MAP ERG pathway by IP3R1 gene, influencing neuronal migration or projection. The JNK pathway was also downregulated in Cntnap2 deficient excitatory neuron. In the case of selected DMs, right here, increase in the PC and SM can be explained by a PC-SM conversion process, but no enzyme involving in this conversion process was found among the DPs, and the potential mechanism of upregulated lipids in deactivation of MAPK, which leads to enhanced PC biosynthesis, as previous study by Dr. Agastian shown that the inhibition of ERK suppressed PC biosynthesis and reduced cellular PC level. To conclude, we performed various integrative multi-omic analysis, trying to better understand what the autism spectrum disorder underlying molecular network is. And we provided a hypothetical pathways, molecular pathways. And I want to give thanks to Professor Min-Si Kim and Kwang-Pyo Kim from Kyung University, Bo-Young Kim in Kyung University, and Yong-Suk Lee in Seoul University, along with Dr. Jiwon Park and Dr. Khan Park. And thank you for your attention, and I'll take questions.